Immunology Insights, Basics and Beyond, Chapter 45, Immune Checkpoint Inhibitors. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored CAR T-cell therapy, a revolutionary way of engineering T-cells to hunt down cancer with synthetic precision. Today, we turn to a different but equally transformative approach: immune checkpoint inhibitors. This time, we are not adding anything new to the immune system. Instead, we are removing something, specifically the brakes that tumors exploit to shut down immune attacks. These brakes, or checkpoints, evolved for good reason. They prevent runaway immune responses, protecting us from chronic inflammation and autoimmunity. But in the context of cancer, these same safeguards can become vulnerabilities. By releasing them, we unleash the body's natural T cells to mount attacks against tumors that were previously invisible. So let's talk science. Let's start with the concept of immune checkpoints. The immune system is finely balanced between activation and regulation. Checkpoints are inhibitory molecules on immune cells that dampen responses once a threat is cleared. Without them, every infection could spiral into uncontrolled inflammation or autoimmunity. Two of the most clinically relevant checkpoints are: first, CTLA4, so cytotoxic T lymphocyte antigen 4. It is expressed on T cells and competes with CD28 for binding to B7 molecules on antigen-presenting cells, or APCs. By outcompeting CD28, CTLA4 reduces co-stimulatory signaling, effectively putting a cap on early T-cell activation in lymphoid tissues. And second, PD-1, so programmed cell death 1. Found on activated T cells, PD-1 engages with PD-L1 or PD-L2, so PD-ligand 1 or PD-ligand 2, on tumor or immune cells in the tissue microenvironment. This sends an inhibitory signal that reduces proliferation, cytokine production, and cytotoxic function of T cells, often right at the tumor site. Well, how do tumors exploit these checkpoints? Tumors are opportunists. Many cancers overexpress PD-L1, sending a biochemical stand-down order to nearby T cells. Others foster a suppressive microenvironment, recruiting regulatory T cells and secreting cytokines like IL-10 and TGF-beta, which further enhance checkpoint signaling. This means even if the immune system initially recognizes the tumor as abnormal, the cancer creates a cloak of tolerance around itself, effectively weaponizing the body's own immune brakes to avoid destruction. Let's have a closer look at the checkpoint blockade and how it is a therapeutic idea. The brilliance of checkpoint inhibitor therapy lies in its simplicity: block the brakes. Here are some examples. First, anti-CTLA4 antibodies, for example used in ipilimumab. They prevent CTLA4 from binding B7, prolonging co-stimulation and sustaining early T-cell activation. Second, anti-PD-1 antibodies, for example used in nivolumab and pembrolizumab. They block PD-1, restoring T-cell effector function within the tumor microenvironment. And third, anti-PD-L1 antibodies, for example used in atezolizumab and durvalumab. They prevent tumor PD-L1 from engaging PD-1 on T cells, keeping the attack switch on. Unlike chemotherapy, which indiscriminately kills dividing cells, or CAR T-cell therapy, which requires complex cell engineering, checkpoint inhibitors leverage the patient's existing immune arsenal. They simply release its restraints. Of course, there are many applications that have quite an impact. Checkpoint inhibitors have redefined cancer care. Here are some examples where checkpoint inhibitors revolutionized therapies. First, melanoma. Once a diagnosis with a median survival of less than a year, it now has patients alive and disease-free more than a decade after therapy. Second, non-small cell lung cancer, or NSCLC. There are dramatic improvements in patients with high PD-L1 expression, turning a uniformly fatal disease into one with long-term survivors. Third, Hodgkin lymphoma, particularly responsive due to frequent PD-L1 overexpression from genetic alterations. Fourth, renal cell carcinoma, head and neck cancer, and urothelial carcinoma. There are significant gains in survival and durable responses thanks to checkpoint inhibitors. As of today, checkpoint inhibitors are FDA approved in over 15 different cancers, and ongoing trials continue to expand their reach. Let's have a closer look at biomarkers for a successful response. Not every patient responds successfully to checkpoint inhibitors, so biomarkers help guide treatment. Here again are some examples. First, PD-L1 expression. Tumors with high PD-L1 are more likely to respond, though responses can also occur in PD-L1 negative tumors, highlighting its complexity. Second, tumor mutational burden, or TMB. More mutations mean more neoantigens that T cells can recognize, increasing the likelihood of immune targeting and a success with checkpoint inhibitors. Third, microsatellite instability high, or MSI-H. Tumors with mismatch repair defects produce a flood of abnormal peptides, making them highly immunogenic. These cancers respond dramatically well to PD-1 blockade. These biomarkers illustrate a key principle. Checkpoint blockade works best when there are abundant, visible targets for T cells to attack. Unfortunately, there are also side effects. So let's look at the immune-related adverse events, or IRAES. Checkpoint inhibitors lower immune tolerance, which can backfire as the immune system starts targeting healthy tissues. Examples include: first, dermatological side effects like rash, pruritus, and vitiligo, which sometimes is a marker of antimelanoma activity. Second, endocrine side effects like thyroiditis and hypophysitis that can cause long-term hormone imbalances requiring lifelong replacement. Third, gastrointestinal side effects like colitis, diarrhea, and abdominal pain due to gut inflammation. And fourth, pulmonary side effects like pneumonitis, a potentially life-threatening lung inflammation. Most IRAES are manageable with corticosteroids and other immunosuppressants, but severe cases may require discontinuation of therapy. Importantly, many patients accept these risks in exchange for the possibility of durable cancer control. Of course, checkpoint inhibitors have a high clinical relevance. Checkpoint inhibitors represent a paradigm shift in oncology. Rather than directly killing tumor cells, they reawaken the patient's immune system to do the work. Their impact includes extending survival in cancers once considered untreatable, expanding therapeutic options across tumor types, and inspiring new strategies. Checkpoint blockade is now tested in combinations with CAR T-cell therapy, cancer vaccines, radiation, and targeted drugs aiming for synergy. So checkpoint therapy has also provided deep biological insight, proving that effective anti-tumor immunity is possible if inhibitory signals are neutralized. To conclude, immune checkpoint inhibitors demonstrate that sometimes the best way to help the immune system is simply to get out of its way. By lifting the brakes, these therapies have given countless patients years of life they otherwise would not have had, and in some cases, genuine cures. The story of checkpoint inhibitors also reminds us that biology often holds solutions hiding in plain sight. The molecules that once seemed like obscure regulators of T cell activity became the basis for an entirely new class of drugs. Looking forward, we can expect further refinements in biomarker-driven therapy, smarter combination regimens, and perhaps new checkpoint targets beyond CTLA4 and PD-1, PD-L1. In our next chapter, we will meet James Allison, one of the pioneers who made checkpoint therapy possible and whose work has inspired an entirely new era of cancer treatment. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time and thank you for listening.